Hi guys, how are you? Today our topic is channel. Channel, what actually channel is? Channel is a specialized transmembrane protein. We can also say pore forming protein, which transports ions and water, etc. along the cell membrane through simple diffusion. And channel is mainly involved in facilitated diffusion. Means the diffusion in which proteins are involved is known as facilitated diffusion. And channel and pump, these both have two distinct features. Channel work along the concentration gradient, but pump work against the concentration gradient. Pump is a specialized transmembrane protein which works against the concentration gradient. For example, if potassium, sodium, anything is coming against the concentration gradient, we will use here pump. Pump will pull the sodium and potassium against the concentration gradient. So it is a specialized transmembrane protein. Now there are mainly two types of channels: gated channel and non-gated channel, or we can also say leaky channels. Leaky channel, they don't have gates, means they allow full flow of ions throughout the membrane. Means they are mainly involved in facilitated diffusion. Means then they have no need of any ligand or any voltage change or any mechanical pressure. So they are known as leaky channels or non-gated. And gated needs some external stimulus. So there are three types of gated channels: mechano leaky channel, ligand leaky channel, or voltage leaky channel. Or we can also say like that: mechano gated channel, ligand gated channel, or voltage gated channel. So let's start from ligand gated channel. Ligand means chemical. Ligand is a chemical which is needed to open the channel. So this is structure of ligand gated channels. It is first divided into subunits, and here is the structure of one subunit. One subunit has two domains. One is extracellular domain and one is transmembrane domain. It is wholly present inside the cell membrane, and the extracellular domain goes out of the cell membrane. It also have intracellular domain which goes inside the cell. So look at the extracellular domain. So here is a site for ligand to bind. So this site is known as ligand binding site. So when ligand binds here, many different type of contractions occur in the transmembrane domain, and as a result, this domain opens and flow of ions starts. Example of this is sodium ligand leaky channel, which is present at the neuromuscular junction of post neuron. Now mechano leaky channel. Mechano is the word mean mechanical. So mechanical pressure or mechanical touch. If we apply touch, so means we apply pressure on the body. So at that time, a channel is used which produces nerve signals. Now look at the image on the screen and look into the center of the channel. If we apply pressure on this, so it will gain pressure and will change its shape. So now look at a second image, it will become like this. It will expand and will allow the flow of ions. So after that, the inactivation occurs, and the inactivation gate from side comes and blocks the channel. After that, when the pressure is released, the inactivation gate comes to normal, and also the channel squeezes and the ion flow stops. Now come to voltage gated channel or voltage leaky channel. Let's look at the screen. Each pump has subunits. Mostly there are 4 subunits in each pump, and every subunit is divided in domains, in 2 domains mainly. So there are 2 domains. The large bigger one that you are seeing, this one domain is called pore forming domain, and the smaller one, this is known as voltage sensor domain. This is mainly sensitive to voltage. Now there are different parts of the pump. So for example, the first part is that one. This is the selectivity filter, or we can also say it's selectivity gate. And this one is the activation gate, or we can also say it voltage gate. And this one, there are types N and C type channels. So in N type, this one is inactivation gate, and this one and this one. And in C type, inactivation gate is present inside the activation gate. Now come to N type and C type. N type is in what do you understand from N? N means nitrogen and C means carboxyl. There are two groups. Nitrogen means amino group and carboxyl group in protein. So in N type, the nitrogen group or amino group is unbonded, we can say, or free nitrogen group, or and C type has free or unbonded carboxyl group. So mainly C type is slow and N type is fast. They both have also different structures as I discussed above. So look at the structure. This structure is of N type. N type, these are this is outer side of the cell and this is inner side of the cell. Inner side is negative and outer side is positive. So when selectivity gate mainly selects the ion, so selectivity gate first selects the ion, and after that the ion passes through the activation gate. So when the membrane is depolarized and it reaches till the threshold, so the negativity decreases on the inner side and the positivity also decreases on the outer side. So as the negative and positive attracts each other, so when the negativity decreased, they start repelling, means the attraction between the negative and positive decreased, so they start repel each other. Sorry, I forgot about that. Mainly the voltage sensor is positive charged, so when they repel each other, the voltage sensor goes up. So as it goes up, the activation gate opens. So the activation gate opens and the ion comes in. And as inactivation gate is a voltage sensor, so at that time when voltage sensor goes up, the inactivation gate starts slowly closing. And as very few ion comes in, so the inactivation gate gets closed. So this is why the N type is also known as fast gates. They closes very fastly, opens and closes very fastly. And after that, when repolarization occurs and the membrane potential is retained to -70 or whatever it is, so and they start pulling each other as the negative charge regains, so the positive and negative attracts each other, so this attracts and the voltage sensor gets down, and also the inactivation gate goes to its original position, and the voltage gate also closes by the pull of voltage sensors. So the N-type potassium channel example is sodium voltage gated channel. Sodium voltage gated channel is mainly the N-type, which we say it is the fastest over potassium and calcium. So it is N-type. It is an example of N-type inactivation gate. Now comes to C type, as it is slow. So look at its structure. Its structure is same, but the main difference is only in the inactivation gate. Here inactivation gate is not present separately. It is present inside the activation gate. Means some part of activation gate inactivates. Its example is potassium voltage gated channel, which is a slow channel. So what is its mechanism? Its mechanism is mainly same, as inner side is negative and outer side is positive. When the negativity decreases, so they repel each other and the positive goes upward. As the positive goes upward, as the activation gate was pulled, so it opens and the potassium rushes in. Potassium rushes in, and as this is selectivity gate which selects only potassium, so potassium goes out, and after when the membrane is repolarized, so the negative and positive will attract each other and the selectivity center will come down again and will push the voltage gate or activation gate, and it retains its original position. So here, the inactivation gate closed directly before repolarization, and here the activation gate closed after repolarization. Here, one concept which I got that it must be important: threshold frequency. You all know what is threshold frequency. The frequency is the minimum amount of frequency at which the activation gate opens. So subthreshold and suprathreshold is the word means sub. So the frequency which is lower than threshold at which the activation gate doesn't open, and suprathreshold which is higher, much higher than threshold frequency. So when the frequency is increased and it reaches suprathreshold frequency, so only the frequency increases at that point. The frequency of the overall graph increases, not the amplitude. The amplitude remains same every time because the depolarization can't exceed from a specific limit. Means, for example, it reached 35 or 40 or some value, so it will remain at that value. It will not increase from that value. Now, there are two types of inactivations, means inactivation states. One is open state and one is closed state. Open state. Open state normally occurs in strong depolarization, and closed state normally occurs in weak depolarization. In closed state, first the channel is closed, and after that it is directly inactivated after depolarization. And in open state, first the channel is closed, then it opens, and after that it inactivates. As we discussed here, first it was closed here, it opened, and after that the inactivation gate closed it. Or in this one, first it was closed, it opened, and after that it again closed after repolarization. So if there are strong depolarizations, so open state inactivation occurs. And if there are very weak depolarization, so what actually occurs there? The gates are repelled very weakly and very slowly, and this very slowly repulsion doesn't pulls the activation gate to open, and as a result, the gate is inactivated before opening. And in this, they also goes very slowly. So also the activation gate closes very slowly, and till the activation gate opens, the inactivation gate comes near to closing. So there is no time between opening of activation gate and closing of an activation gate.